Welcome to the Nature of Teaching Professional Development webinar series. My name is Rod Williams, a professor and extension wildlife specialist with Purdue University Extension. Today's webinar is entitled Disease Ecology, Part 1. This will be a 30-minute webinar covering the principles of disease ecology and the role of parasites in natural ecosystems. If you're unfamiliar with disease ecology, this webinar will provide you with science-based background information to prepare you for the webinar on disease ecology, Part 2. Well, joining me today is a good friend of mine and colleague, Dr. Jason Hoverman. Jason is an associate professor here at Purdue and a co-author of the unit on disease ecology. Jason's going to be highlighting key concepts and challenges in the field of disease ecology, and then I'll finish up the webinar highlighting additional resources and the procedure to obtain your certification of completion. So Jason, at this point, I'll just simply turn things over to you and you can get us started.
All right, thank you very much for the introduction, Rod, and thank you all for joining us today. So a little bit of an overview about what I'll be talking about today. So I'll cover first disease ecology and parasites, give you a little bit about the basics of disease ecology, and then we'll move into the significance and importance of parasites and really underscore to students, hopefully, that these are really important components of our ecosystems. And then I'll talk a little bit about the diversity of parasite species, disease modeling, and also go into a little bit about amphibian parasites. And these last three feed into our lesson plans that we've developed for you. So let's start off with disease ecology and parasites. So what is disease ecology? So this is this ecological study of host-parasite interactions within the context of their environment as well as evolution. Okay, so let's dive a little bit more into what this actually means. And disease ecology really focuses on the disease triangle. So it consists of host, the parasite or pathogen, as well as the environment. So all three of these things are critical when it comes to understanding disease dynamics. So in particular, if we alter any of these three aspects of the disease triangle, this influences maybe the prevalence of a pathogen or the outcome of disease. So I'll give you an example of this. So let's say an environmental challenge comes in, say it's a contaminant, and that contaminant increases the susceptibility of a host to a pathogen. So that can increase disease risk for that overall host and the population as a whole. So again, the disease triangle is really trying to bring together these three aspects to understand disease dynamics in nature. And also, I just want to briefly talk about what an actual parasite is, right? So these are organisms that live in an intimate relationship with a host of a different species and have a detrimental effect on that host. So in other words, they're going to reduce the fitness of that host species. So this could be the survival, the reproduction, or even just the body quality of that individual. And over here on the right, what you'll see are a broad diversity of terms that we use to describe parasites. So I'll most often use parasite or pathogen kind of interchangeably, but you also see terms like macroparasite, microparasite. In birds, there's examples of what we call brood parasites. So these are birds that lay their eggs in the nest of another species. And there's also parasitoids. So these are parasites of insects. Typically, they're wasps that lay their eggs inside insects. And a really fascinating example, which are hyperparasites. These are actually parasites of parasites. So really fascinating examples in this realm of parasitology as a whole. So again, broad diversity of terms. In the teacher notes, we'll have some descriptions of these different categories of parasites. But again, I'll mainly use pathogen and parasite kind of interchangeably today as I go through this webinar. And also in the teacher's notes, we provided a broad diversity of terminology that's used in disease ecology. So like any field, we're using lots of different terms. So we've defined all those terms for you, including things like host types, disease terms, as well as transmission, what that means. So please refer to those teacher notes to get a little bit more information about those. And I'll come back to this one a little later on when we get to the amphibian parasites, so the types of hosts in disease ecology. So with that broad introduction to disease ecology and parasites, what I want to do now is jump into the significance and importance of parasites. This is something I like to underscore to students because we typically don't think about parasites playing very important roles in ecosystems. So again, I'm going to try to underscore that to you guys, and hopefully you can transfer that to your students as well. So of the many questions I get about parasites, one of the main ones is exactly how many are there? And this is a really challenging question. You know, obviously these things are really small. They end up living within many of their hosts. Also, there's limited training. So there's not very many people that have the expertise and the skills to identify all the different types of parasites that might be in a given region or might be infecting certain hosts. And we also have a pretty strong bias in terms of geography as well as host. So from a geographic standpoint, we tend to focus on places like North America, in the United States, maybe Europe and Asia, and we don't focus as much in the tropics. From a host bias standpoint, we tend to focus on mammal parasites because we ourselves are mammals. We tend to be more interested in those things that could possibly infect us. So as a consequence, we don't know a lot about, say, fish parasites or parasites of plants in some cases. So given those challenges, is there some way we could potentially estimate parasite diversity? So let's think about that a little bit. And over here, what you're seeing here is the total described species on the planet. So we're down to about, or up to about 2 million, 2 million parasite species, or not parasite, total species that have been described currently. And it's estimated that we could have even close to 11 million species on the planet. So if you just do a simple thought exercise and think about, well, if every free living species has at least one parasite species that's specific to it, that would mean that roughly 50% of total biodiversity could be parasitic in nature. And that's not a really crazy statement if we think about this figure right here. So this is the average number of parasites per host for a bunch of different groups. This actually comes from a general ecology book. And you can see for the vast majority of these groups, they have way more than one species that infects them. So this isn't that crazy in terms of an overall estimate of parasite biodiversity. So that means we're around maybe 1 to 5 million parasite species at a minimum on this planet. Okay, so there's lots of them. There's broad diversity of different types of parasites as well. Okay, so what? There's lots of them. What can they do? Okay, so one of the important things they can do is actually regulate host populations. And historically, we've generally thought about maybe competitors or predators playing the important role in regulating populations. It wasn't until the 80s that we figured out that parasites can do a very similar thing. So in the 80s, a study was done with red grouse. This is in the United Kingdom. And for these red grouse, what they noticed from hunter-harvest records going back 100 years are these dramatic fluctuations in the population size of these birds. And what they started to think about was, well, there's not many predators in this system, but these birds are infected with a worm. So in this case, a nematode. And this nematode was very common in these bird populations. So in the 80s, researchers got interested in figuring out if these parasites are actually regulating the bird populations. So they did some experiments. And they had a control population in which they just left it unmanipulated, and these birds still had their parasites. And what they saw were these classic fluctuations in the population size. But then they also took populations and they cleared them of their parasites using a drug. And what they found is this completely eliminated the population fluctuations. So again, this is in the 80s, so not that long ago. And this is actually demonstrating that this parasite regulates this host population. Okay, again, first demonstration of this. And since then, a large number of studies have come out demonstrating that indeed parasites can play an important role in regulation. In addition, we also know that parasites have shaped human evolution, as well as the evolution of many host species, right? Think about our innate as well as adaptive immune systems. These systems have evolved to fight off various types of parasites and pathogens, these challenges to the body. And if we think more broadly, parasites are also extremely important in food webs. And so provide an example from you. This example comes from estuary systems in California. So what these researchers did is went to the estuary system and they looked at the food webs in these systems. So what you're seeing on the right here is just your basic food web diagram for the free living species. So you have trophic level going up here on the y-axis and just free living species going here on the x-axis. So it looks pretty well connected. But this is in the absence of parasites. And this is what happens when you start to include all the parasites that exist in this estuary system. So you can see a large number of connections being made across all these different free living species. So what this is demonstrating is that parasites play an important role in enhancing food web connecteds. And the reason for this is many of these parasites have complex life cycles. So they're using multiple host species and connecting up all these species within the food web more dynamically. And also from this same study, what they looked at was basically who's eating who when it comes to the natural enemies in these systems based on the trophic level. So in this diagram, what you're seeing here is on the x-axis, this is basically the trophic level going from basal trophic level 1 all the way up to the apex at trophic level 6. And what they're doing here is showing you the natural enemies per consumer. So if you have predators, that's in the dark part of the bar. And then if it's a parasite, it's in the white portion. But what you'll notice here is as you move up the food chain, up the food web to the highest trophic levels, those apex predators are being consumed by actual parasites. So in this case, we might actually think about parasites being the apex predators in these systems, where historically we thought, maybe that's a bird or a large fish. But actually those top predators are being consumed by parasites in these systems. So again, just underscoring that parasites play an important role across levels of biological organization. Okay, so now what I want to do is just dive into the diversity of parasites. So we've talked about how many parasites there are. Let's talk a little bit about just how diverse parasites are. So what you're seeing here is just some major parasite groups going from viruses to fungi, all the way down to arthropods, including things like mosquitoes that can vector other types of pathogens as well. Now, I'm not going to go through all these groups. I'm just going to provide a few examples of some of the well-known kind of pathogens and kind of currently pathogens of current interest. And then we'll talk about the lesson plan we've developed around students getting to familiarize themselves with the diversity of parasites. So let's start with a virus. In particular, thinking about Ebola viruses. Okay, this is a group of viruses actually in the genus Ebola virus. Excuse me. They cause hemorrhagic disease in humans as well as primates. And what's really interesting about this, it's relatively new to science. So they were discovered in the 1970s, and then since then, there's been about 24 sporadic outbreaks of Ebola virus. And most recently, I believe it happened in 2014 in Africa. And this actually caused a global scare. There's lots of concerns about this spreading from Africa, moving to other continents. So there's lots of concerns about shutting down airports and restricting movement of humans to prevent the spread of this across the world. Now, luckily, it didn't necessarily move out of Africa, but this is still something that Africa is kind of dealing with, even currently, 6 years later, with that current outbreak. But this is also an ecological problem. So going back to disease ecology, there's some evidence suggesting that people going into the forest, collecting bushmeat, so that's what you're seeing here. So they're collecting animals to sell in markets for fresh meat. That seems to play a role in whether or not transmission can occur. But also human interactions with bats. Bats appear to be a reservoir for Ebola virus, so you see one here. So in coming to close proximity to colonies of bats, that can lead to spillover to human populations. And again, if you're going out looking for this bushmeat, you're more likely to encounter these bats, and that might lead to transmission. So it's a very interesting ecological problem in trying to predict when these outbreaks of Ebola virus might actually occur. Another well-known example of a disease is Lyme disease. So this is caused by a bacterial pathogen, Borrelia. And this is something that originated in the northeastern United States. As you see over here, this is showing you basically the spread of Lyme disease over time. So it's basically making its way out of northeastern the US. And then it's basically jumped over here to kind of the north central region as well, into Wisconsin, coming down into Illinois, and starting to be picked up here in Indiana as well. But again, this is another ecological problem because we have this pathogen that's being vectored by ticks. And these ticks use a variety of hosts that complete their life cycle. So key players in this life cycle include things like mice and other rodents, white-tailed deer, which can harbor large populations of ticks, and of course humans that come in contact with all these animals. Maybe they're walking through a forest, on a hike. This can lead to spillover to the human population, and indeed, this is why we're seeing much of this spread in the United States currently. So again, another kind of interesting example of a pathogen that kind of touches home for many of us here in the United States. Okay, now I'm going to provide an example of a more wildlife-focused pathogen. And this is the white nose syndrome. So it's caused by a fungal pathogen, Pseudogymnoascus destructicans. And this pathogen was thought to be introduced from Europe or Asia. And it's basically colonized the bat populations in Eastern North America. And what it does is it grows around the muzzle of the bats, as you see here. And it also will grow on the membranes of the skin, for the wings. And what happens is it basically depletes the resources of these bats. And as a consequence, especially when they're hibernating over the winter, they actually will waken during the wintertime and try to go out and forage. And when they do that, they can die from exposure if the temperatures are too low and there's maybe snow on the ground and they can't find their resources. So unfortunately, this has led to a decline in many bat populations, including the Indiana bat, an endangered species. So it's a big concern, and it's actually jumped not just from the eastern US here, but it's jumped over to the western United States as well, and has colonized some bat populations out there. So there's a lot of concern about bat populations and this white nose syndrome. Okay, so the final example I have for you is just a really fascinating example. And this is a complex life cycle parasite called an Acanthocephala. So it's also called a spiny-headed worm. So you can see a small picture here of it here. And it gets its name because of all these spines on its proboscis. And what it does is basically lodges itself in the gut, the intestinal wall of its host. So what you're seeing here is one of its hosts. This is an amphipod. And this bright red spot right here is the Acanthocephalan parasite. Now, as I mentioned, this is a complex life cycle parasite. So it actually has to end up in a fish to complete its life cycle. So by inducing this red spot or forming this red spot in the amphipod, it makes the amphipod stand out more to the host, the fish. And the fish is easier to find this amphipod and consume it, and therefore that completes the life cycle of the parasite. So a really fascinating example of induced vulnerability to predators caused by a parasite. Okay, so this brings us to the lesson plan. So what we've developed is this idea of thinking about parasite diversity. And what students can do, you can break them into groups or have them do this individually, is just do a basic internet search on parasites that you assign to them. So you can do that search and look for things like the disease that's caused by that parasite, the host range, the life cycle, the geographic range, other random facts they can find about that parasite, and they can present that to the entire class. So this is a really quick and easy way to learn about parasite diversity and just the fascinating world of parasitology as well. Okay, so this brings us to disease modeling basics. And I like to introduce this to students because oftentimes when they hear modeling, they get kind of scared. But what I hope I can kind of instill in you guys, you can transfer this to your students, is that this can actually be a really fascinating way to think about nature in general. So why model disease dynamics? I won't go through all of these, you can kind of read through them, but there's lots of various reasons, as you see here, for why we want to do this. So some of them include, this helps formulate hypotheses about how we think a pathogen might be operating in a host population. It also forces us to come up with assumptions of how we think a disease might be operating. And once we run a model, this can help us determine appropriate management efforts, right? So if we need to cut transmission rates, if we need to vaccinate a certain proportion of the population, that modeling effort helps us guide our efforts. And then lastly, in many cases, modeling is all we can do. For some species, including humans, we can't necessarily do experiments. So it's either logistically or ethically impossible to do that. So we can use these models as a surrogate for how we think, again, a host pathogen interaction might play out in nature. So the model I'll talk about today is based on this dynamic of infection within a host. And just, you don't need to focus on all of this. Just kind of walk you through what's being shown here. So what you're seeing is the time course of infection in a given host. And what you'll see in this kind of shaded area, this is basically infection within the host. So this is the pathogen growth within the host. So you can see from the time of initial infection that pathogen ramps up, the population size increases, and then eventually it goes down. And the reason it's going down is what you see at this black line right here. This is the host immune system. As the infection ramps up, you see that immune system ramping up as well. And when that immune system peaks, that basically clears the pathogen from the host. So what our model is trying to do is capture these different dynamics. So it's trying to capture, in particular, these three categories. The time point when the host is susceptible to the infection, the time point in which it gets infected and becomes infectious, and then the time point in which it's recovered from that actual infection. So this is what we call an SIR model. So susceptible, infectious, recovered. So we're trying to take those dynamics within an individual host and scale it up to a population level. So this is what we call a compartment model as well. So just kind of simplify this. We have susceptible. These are individuals that can catch the infection. The infectious have it and they can give it to the susceptibles, as you see this dotted line here. And then the recovered are able to clear that infection. So again, very simple compartmental model. Now, obviously, with all models, there's lots of assumptions. Again, I won't go through all of these assumptions here, just highlight a few. The first is that the population is fixed, so there's no birth or death in this model. Also, it's unidirectional flow. So individuals can only go from susceptible to infectious, and then from infectious to recovered. And then lastly, I'll just highlight there's no variation within the population. So there's no variation in age, sex, size, anything that would influence your probability of getting infected, essentially. So again, you can go through this with the students in terms of these assumptions and what they actually mean. But just kind of thinking about the dynamics of this model, again, these are the equations that we use. And what you have here is beta. This is the transmission coefficient. Gamma, which is the recovery coefficient. So in this case, what we see is that susceptibles, again, the only way you can move from this category is to get infected. So they're only going to lose individuals. Those individuals come down here into the infectious category. And then they're going to lose individuals based on the recovery rate. And those individuals add to the recovery category. So again, this unidirectional flow moving within this model. So what this would look like if you actually graphed it out is shown here. So what you see is the proportion of the population that fall within each of those categories I just mentioned. So this is the susceptible group in the population. The dotted line here is the recovered group. And this line right here is the infected group. So essentially, this is the epidemic right here. So the height and the breadth of this are basically characteristics that we use to describe the epidemic. So if you had more infected, obviously the epidemic would peak up here and come down. But in this particular example, based on the parameters that were used, what you'll notice is we actually don't infect the entire population. Okay, so some individuals never get the infection, and about 60% of them do get the infection and recover from that infection. What's really interesting about this model as well is that you can alter it how you see fit based on the disease that you're working with. So for example, what you could do is take out long-lasting immunity. So take out the recovered category in this case, and you just revert to what's called susceptible infectious model, so an SIS model. So this would be ideal for something like the common cold, something that we can always pick up. Anytime this comes into our communities, we tend to pick up the common cold. Or you could add something called a latent or exposed period. So in this case, once you become infected, you move to this exposed category. So when you're in this category, you actually cannot infect anyone else. It's not until you move to infectious that now you can have infection spreading to susceptible category. So an excellent example of this is chickenpox. So there's about a 10 to 21 day latent period or exposed period where individuals are not infectious, but that infection is ramping up within that individual host, and then they transition to the infectious category. So with your students, you can think about ways to alter these models to capture different types of host pathogen interactions. Okay, so the lesson plan we've developed was basically geared towards students visually seeing how disease progresses in a population using the SIR model. And we came up with two basic approaches for doing this. One's called the bingo chip approach, and the other is a computer simulation. So with the bingo chip approach, what students will be doing is working with cups, and they had different colored chips that represent these different infection categories: susceptible, infected, and recovered. And they move these chips based on certain rules, and what they're able to do is, again, model the disease dynamics based on the rules that they formulated for that particular pathogen. And then another way we wanted to visualize this is actually through computer simulation. So this gives them really kind of real-time data that they can alter the parameters in that model and see how that alters disease dynamics. So in particular, we'll have spreadsheets, computer spreadsheets, very simple Excel spreadsheets, for example. They alter the parameters, and they're going to focus on this epidemic spike here. Okay, so how high is that spike? You know, how wide is that spike? So these are all developed in the lesson plan for the students. Okay, so this brings us to the last component, which is talking about amphibian parasites. So this particular activity is going to be a little bit more hands-on for the students. And just provide a little bit of context here. So I've been working with amphibian parasites for about 15 years or so, and it's amazing the broad diversity of parasites they get infected with. But what we'll focus on, though, are trematodes. These are parasitic flatworms that infect amphibians. So you just see a variety of species here, so you can see some of the differences in kind of body shape and size of the different parasites. So in some cases, these amphibians can be infected with about 15 to 20 different types of trematodes in certain types of habitats. But again, these parasites have a complex life cycle. So what that means is they're using these multiple host species to complete their life cycle. So the first intermediate host is this freshwater snail. And these freshwater snails become infected from the feces of the definitive host, which is typically a waterfowl, a mammal, or a reptile, where the parasites have shed eggs into the feces. Once that feces ends up in the water, those eggs hatch and infect the snail. Now, within the snail, another stage of the parasite develops. It's called a cercaria. And that cercaria emerges from the snail, which you see here, and it seeks out a larval amphibian, so a tadpole. It infects the tadpole and it forms a cyst. Okay, at that point, it's just sitting there waiting for that amphibian to be consumed by a definitive host. And once it is, that completes the parasite life cycle. Okay, so again, these parasites require each of these components in the life cycle or else they can't persist. So just provide a couple of examples of some of these parasites. So the first one I'll talk about is Riboria. This particular parasite is well known because it causes amphibian malformations. So these malformations that you see here, these extra sets of limbs, are commonly what are caused by this particular parasite. And the reason it does this is the parasite drills into the side of the tadpole, and those tadpoles are developing legs. And this parasite, it's relatively large, and it causes a physical disturbance for that amphibian limb. And as that limb grows, it can splinter, and various other more malformations can form as a consequence of that physical disturbance of the parasite in that developing limb bud area. And so what I'm showing you here are the broad diversity of malformations that have been detected with this parasite. So this is from some of my colleagues' work, Peter Johnson out in the University of Colorado, and just showing you some of the crazy malformations that he's encountered over his time. So you see anywhere from missing limbs, here's a case of multiple limbs in a tadpole, extra limbs again. This is a skin webbing over here. This is where the limb can't fully straighten because of the extra skin developing in those areas. So again, very kind of well-known parasite because of these extreme malformations. And these malformations, they function to increase the chances that the amphibian is consumed by a definitive host, which again is typically a bird in this case. So are less able to get away from that predator. Another parasite that we frequently work with in my lab are the Echinostomes. This is actually a group of parasites, but these parasites actually swim up the cloaca of the amphibian and they settle in the kidneys. So these are actually dissected kidneys from an amphibian. And each of these circles here, if you zoom in, these are all the cysts of that parasite. So in some areas, for example, bullfrogs and green frog tadpoles, they'll have thousands of these cysts in their kidneys. And when they get that high parasite load, you can get something like this. This is body swelling or edema. The kidney plays an important role in osmoregulation. So as a consequence of these infections, these amphibians are not able to clear the water from their body, and they get this body swelling. And this parasite, very common. We find this throughout Indiana, basically throughout the United States, anywhere where we find those three components, the amphibians, the snails, and the definitive host that I mentioned before. So the lesson plan we've developed, the goal here is to get students to learn about how these amphibians can use avoidance behaviors to avoid getting infected with these parasites. So what they'll do is they basically go out into nature, they'll collect some of the snails, they'll do something called shedding, which again is all explained in the teacher notes and the lesson plans, but they'll be able to obtain the parasites from these snails and use those parasites in exposures to tadpoles. And what they're able to do here is basically do activities in the field, bring this back to the laboratory, as well as do experiments. So it gives them basically the feel of being a scientist and exploring, again, how these behaviors can be used to avoid parasites. So with that, I'll end and turn it over to Rod. Thank you.
Great, thanks, Jason. At this point, what I'd like to do is first thank everyone for joining us on the Nature of Teaching Professional Development webinar entitled Disease Ecology, Part 1. I hope you consider participating in the second webinar on disease ecology to find out how to incorporate the unit lessons, many of which Jason just gave you a quick preview on, how to incorporate those lessons into your classroom. Now, if you enjoyed this webinar, I also hope I encourage you to click on the card that should be popping up right now on the top right of your screen. That'll take you to the Nature of Teaching YouTube channel. Here you can view sneak peeks of the lesson plans that are directly related to the webinar that you just watched. So essentially, if the card isn't working for you, I've also embedded the URL of our Nature of Teaching YouTube channel for you to click on, or if you're in YouTube, you can simply search in the YouTube search bar for the Nature of Teaching, and those sneak peeks will pop up. Essentially, all these sneak peeks are a 2-minute or less set of videos that really highlight those exciting activities that Jason talked about in the webinar today and walk you through what those activities are going to be, the lesson plan, the state standards that they might be addressing. Just a great way to give you an idea of what that lesson has to offer. Now, if you're interested in obtaining a certificate of completion for this 30-minute webinar, now you want to click on the second card that should be appearing in the top right of your screen. Here you'll be directed to a short Qualtrics survey that provides us some feedback on the program. Once you complete this Qualtrics survey, you'll automatically be emailed the certification of completion. Jason, if you'd like to rejoin us, I have a quick question for you. First, I want to thank you for sharing your knowledge and expertise with us today. If viewers have additional questions related to disease ecology or the roles of parasites in nature, is there a way they can reach out to you for answers?
Yeah, for sure. So they can contact me via the email that you see here listed on the screen. Yeah, I'm happy to answer any questions and look forward to any feedback you might have as well.
Perfect. And if you have any questions on the procedures to get your certification of completion, you see my email there as well, rodw@purdue.edu. Happy to answer any questions you may have there. So at this point, Jason and I both would like to thank you. We hope you enjoyed learning with us and consider participating in additional professional development webinars offered here by the Nature of Teaching team. Until then, thank you for engaging our youth with nature. Until next time.